Hello everyone and welcome to the second week of online teaching. This week we are going to continue with protein structure. Let's get started. The three learning intentions today are to understand the three types of secondary structure and the bonding within secondary structure, to understand the bonding that takes place in tertiary structures to create a 3D structure, and to understand how multiple subunits and prosthetic groups create the quaternary protein structure. As we discussed in last week's video, there are four main levels to protein structure. There's primary structure, secondary, tertiary, and quaternary. We've already discussed primary structure, which is when a specific sequence of amino acids are joined together by peptide bonds, and this creates a polypeptide chain. We're now going to look at what happens after a polypeptide chain so that we can create a fully functional protein. Secondary structure is the next phase of protein structure. You're required to know the bonding that takes place in secondary structures along with three types of secondary structure. Hydrogen bonding allows the polypeptide chain to be stabilised. It is important to know that hydrogen bonds bind to the peptide bond between the amino acids. They are found along the backbone of the protein strand. When we say the backbone, this just refers to the long amino acid chain or the polypeptide chain. That's all we're referring to. There are three secondary structures that can be visually identified. The first is called an alpha helix, then there are structures called beta pleated sheets, and finally structures called turns. Whenever you see the word helix, it refers to something shaped like a corkscrew. So for example, a double helix is shaped like a corkscrew or like a spiral staircase. An alpha helix can also have this characteristic corkscrew structure. The variable R groups that can determine whether an amino acid is acidic, basic, polar or hydrophobic stick on the outside of the alpha helix, as can be seen in the diagram. So in this diagram here where it says R, where it's sticking on the outside, these are the R groups. Beta pleated sheets have a structure that looks a bit like a corrugated sheet or corrugated iron. The R groups sit above and below the sheet as seen in the diagram. Now there are two types of beta pleated sheets. There are antiparallel and parallel beta pleated sheets. Antiparallel run in the opposite direction to one another, while parallel run in the same direction. This is based on their N and C termini, which we are going to look at in the next slide. Polypeptides have to start and end somewhere. The C terminus is one end and ends with a carboxyl group, while the N terminus ends with an amine group. In a parallel beta pleated sheet, both the C terminus are at the same side and the N terminus are also at the same side. So you can see that in this diagram here. We've got both C terminus on the left hand side, whereas the N terminus are on the right hand side. While in antiparallel beta pleated sheets, the strands run in opposite directions. The C terminus are at the opposite ends as are the N terminus. So you can see the C and N terminus on the left hand side and the C and N terminus on the right hand side. This diagram is a really good representation of what parallel and antiparallel mean. Antiparallel is when the sheets run in opposite directions, and you can see that with the opposite arrows. While parallel, the sheets run in the same direction, so the arrows are pointing the same way. Turns are the third type of secondary structure. They serve to reverse the direction of a polypeptide chain. So if you see something that's reversing or going in the opposite direction, then this is going to be a turn. Okay, so I'm going to give you 5 seconds. Can you tell me which secondary structure this is? Okay, so well done if you got this correct. Region X is an alpha helix, and that's because it's got this corkscrew helix structure. Okay, so we've looked at the first learning intention to understand the three types of secondary structure and the bonding within secondary structure. We're now going to look at number 2 to understand the bonding that takes place in tertiary structures to create a 3D structure. After secondary structure, the polypeptide folds into its final 3D structure, known as the tertiary structure. Folding at this level is stabilised by many different interactions between the R groups of the amino acids. You need to know all 5 interactions that allow the tertiary structure to maintain its shape. The first interaction is between hydrophobic amino acids, like the ones that we looked at last week. These amino acids do not like to be in contact with water, so they tend to stay in the middle of a protein. Hydrophobic interactions then occur between these amino acids, and you'd usually find hydrophobic interactions within the middle of a protein. In an ionic bond, atoms are oppositely charged and therefore held by electrostatic attraction. Ionic bonding is a type of chemical bonding that involves the electrostatic attraction between oppositely charged ions, so positive ions and negative ions. These ions represent atoms that have lost one or more electrons and atoms that have gained one or more electrons. So usually where you'd see an ionic bond is with an atom that has a negative charge and an atom that has a positive charge. So you'd find an ionic bond between them. Hydrogen bonding is initially found in the secondary structure, although we talk about it in the tertiary structure as well because it helps to create that final 3D shape. A hydrogen bond is an electrostatic attractive interaction which occurs between a hydrogen atom and an electronegative atom, such as oxygen or nitrogen or fluorine. So if you see a bond that is between a hydrogen atom and either oxygen or nitrogen or fluorine, this is a hydrogen bond. A disulfide bond, also known as a disulfide bridge, is a covalent bond between 2 thiol groups. And these thiol groups usually have sulfur within them. One final charge effect which influences the tertiary structure of a protein is London dispersion forces. The London dispersion force is a temporary attractive force that results when the electrons in 2 adjacent atoms occupy positions that make the atoms form temporary dipoles. London dispersion forces are weak bonds. They're also termed van der Waals forces, but you now need to know that they're called London dispersion forces. Okay, so this diagram just represents a polypeptide shown in the purple, and then 4 different interactions within the tertiary structure. So the first one that we've got here, a hydrogen bond. Now the hydrogen bond is between a hydrogen atom here and then between an oxygen. And remember we said that a hydrogen bond is usually between a hydrogen and either oxygen or fluorine or nitrogen. Hydrophobic interactions occur between hydrophobic amino acid R groups, and from our last lesson we know that hydrophobic R groups have carbons and hydrogens within them. So we can see that these 2 R groups are interacting, so this is a hydrophobic interaction. The disulfide bridge is next. It's between 2 thiol groups, so we can see there's sulfurs here which are in the thiol groups, so this is called a disulfide bridge. And finally, ionic bonds are between two oppositely charged ions. So we can see that this is a positive ion and this is a negative. So when they join like this, it's called an ionic bond. Okay, so again, I'm going to give you 5 seconds. Can you tell me which bond C is? Well done if you've got this correct. C is a disulfide bond, as it is between two sulfur thiol groups. Okay, I'm going to give you 5 seconds. Can you tell me which bond B is? Well done if you've got this correct. B is a hydrophobic interaction. We know this as it's between two hydrophobic R groups. Remember, hydrophobic R groups are composed of carbon and hydrogen atoms. Okay, can you tell me which bond A is? Okay, well done if you've got this correct. A is a hydrogen bond, as it binds between a hydrogen atom and an electronegative oxygen atom. Okay, so finally, can you tell me which bond D is? Well done if you've got this correct. D is an ionic bond, as the positive nitrogen in the amine group attracts the negative oxygen in the carboxylic group. Okay, so we've looked at to understand the bonding that takes place in tertiary structures to create a 3D structure. Now we're going to look at the final learning intention to understand how multiple subunits and prosthetic groups create the quaternary protein structure. Many proteins are made up of more than one subunit. Quaternary structure exists in proteins with several connected polypeptide subunits. The subunits are linked by bonds between the R groups of the polypeptide chains. For example, hemoglobin is made of 4 subunits. This can be seen by 2 blue subunits, so 2 blue subunits in the diagram, and then 2 red subunits in the diagram, so a total of 4. Some proteins include prosthetic groups or non-protein groups. A prosthetic group is a non-protein component that's tightly bound to a protein and it's necessary for the protein's function. An example of this is the heme group, which is bound into hemoglobin. Now we know that hemoglobin is an essential part of a red blood cell. It carries the oxygen around the body. In hemoglobin, we have a heme group, which is our prosthetic group, and that contains an iron atom. And the iron atom is the site of the oxygen binding, so that's where oxygen will bind onto. The iron atom is covalently bound to the hemoglobin via a series of histidine amino acids, and because it's bound, it allows the oxygen to bind into the whole hemoglobin component and be taken round the body. Now we know all 4 protein structures. This forms a good picture of how proteins go from being synthesized at the ribosome to being fully functional. Primary structures are polymers. They are long polypeptide chains consisting of a sequence of amino acids joined by peptides. You can see that here in the diagram. We've got a long sequence of amino acids, and each of these amino acids are joined by peptide bonds. Hydrogen bonds help to fold the protein into secondary structures, including alpha helices, beta pleated sheets, and turns. So we can see that here in the secondary structure. We've got an alpha helix here. These arrows represent a beta pleated sheet, and this here is a turn. Tertiary structures then have multiple interactions to create a final 3D structure. So we have got our hydrogen bonding from our secondary structure. We've also got ionic bonding, hydrophobic interactions, London dispersion forces, and disulfide bridges. Our final protein structure is quaternary structure. This is when there's multiple polypeptide subunits. We can see that here. We've got 1, 2, 3, 4 polypeptide subunits. And then if you have a wee look at these grey components here, these are known as prosthetic groups. They are non-protein components, but they're essential to the protein's function. There are two factors that can affect protein structure. Just like in enzymes, the two factors are pH and temperature. As we know, enzymes are made of protein, so this is why that's linked. Temperature and pH affect the interactions holding the protein together. When pH is at its extremes or temperature is too high, the interactions will break, causing the protein to lose its structure. We know that a protein losing their structure to where they cannot be repaired is known as denaturation. Okay, so just to recap, the 3 learning intentions today were to understand the 3 types of secondary structure and the bonding within secondary structure, to understand the bonding that takes place in tertiary structures to create a 3D structure, and finally to understand how multiple subunits and prosthetic groups create the quaternary protein structure. Now that we have went through the PowerPoint, these are the revision tools we are advising you to use. The summary notes can be found in the Files section of Teams. If you look at pages 31 to 33, this gives you all the information you need this week about proteins. I've also attached scanned versions of a revision book for you to read. Finally, if you type in Study Stack and in the search bar type St Ninian's HS, this will come up with loads of flashcards. Click on the Advanced Higher Proteins link to get flashcards for the topic we just covered. This week we have 2 assessment materials for you to complete. The first are revision questions for you to download and complete. If you cannot do this, write these on a sheet of paper for me. The second is a proteins quiz on Teams. This will be put up on Thursday. Once you've completed this, you must submit it. This will show us that you've completed the task. Okay, thanks very much.